We looked at different types of cells and different psychedelics and found that they were all anti-inflammatory. Even LSD, which was the least potent, was still as potent as a typical corticosteroid.
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to SmartDrugSmarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to SmartDrugSmarts. My name is Jesse Lawler. I am your host, and this is the 196th episode of this podcast dedicated to anything and everything that you can do to better help the workings of your own brain, or in certain rare cases, using illegal drugs which are usually intended for the brain to make physical improvements to the body. Yes, this does sound like a weird edge case, and it is one. We're going to be having a really interesting episode today looking at psychedelic compounds, something that we've talked about on numerous previous episodes, but going at it from a different angle this time, examining the little-known fact that in many cases, these substances seem to have anti-inflammatory properties. Inflammation, as you know, implicated in everything from arthritis to physical bruising to some neurological disorders. It's there for a good purpose, but long-term chronic inflammation is definitely a bad thing. Could psychedelics present one possible remedy? That's going to be the subject matter with Dr. Charles Nichols in the main interview. If you hang around for the very end of the episode in the Ruthless Listener Retention gimmick, because it is Labor Day weekend this coming weekend, I know that for many people this is a very alcohol-fueled holiday, and we actually have something both positive and scientific to say about alcohol in the Ruthless Listener Retention gimmick. But for right now, let's kick things off as usual with This Week in Neuroscience.
SmartDrugSmarts, This Week in Neuroscience.
So given that this is Labor Day weekend coming up, I figured it would be good to do a This Week in Neuroscience about work. This is actually from about a year ago that it was published in PLOS One, the Public Library of Science, a study about workaholism from the University of Bergen in Norway. This was a big study. They were looking at workaholism and psychiatric disorders, trying to find out what the correlation there was, if any. The study had some 16,000 plus workers, average age of 37, but the age range was from 16 to 75 years old, so quite a swath. These people were given a battery of tests, including the Adult ADHD Self-Report Scale, the Obsessive Compulsive Inventory, the Hospital Anxiety and Depression Scale, and the Bergen Work Addiction Scale. The long story short is that the correlations between workaholism and psychiatric disorders were positive and significant. If you knew somebody had one of these psychiatric disorders, their chances of being a workaholic improved. If you knew somebody was a workaholic, the chances that they also exhibited a psychiatric disorder improved. Said clinical psychologist Cecile Anderson, taking work to the extreme may be a sign of deeper psychological or emotional issues. Whether this reflects overlapping genetic vulnerabilities, disorders leading to workaholism, or conversely, workaholism causing such disorders, that remains uncertain. Only 7.8% of the people in the study sample were found to be workaholics, but among the workaholics, 32.7% met the criteria for ADHD, a little over 25% for obsessive-compulsive disorder, 1/3 met the criteria for clinical anxiety, and 9% met the criteria for clinical depression. In all cases, the chances of these problems occurring were about 3 times higher than in the non-workaholic segment of the people studied. The Bergen Work Addiction Scale is actually just a 7-question test where you say how strongly you do or don't agree with a statement. Based on those answers, it'll say yes, you are, or no, you are not a workaholic. I will raise my hand and plead guilty. I do categorize as a workaholic based on this test. We will put the 7 questions up for this Bergen Work Addiction Scale at the post for this episode, smartdrugsmarts.com/196. And you can see if you too have triple the regular chances of psychiatric disorders.
SmartDrugSmarts, the podcast so smart, we have smart in our title, twice.
Picked up a couple of 5-star reviews on iTunes. Listener Superheated Samovar from the USA says, always informative, always interesting, and always entertaining. And listener Katarina May, also from the USA, says, I recently did a 14-hour road trip and listening to SDS helped pass the time so very fast, both informative and addicting. So a big thanks to both of you. Thanks also to listener Woody Hulst, who sent me the story that will be the basis for the Ruthless Listener Retention gimmick for this week. Very much appreciate the articles that are sent my way, scientific papers, even weird personal anecdotes about your own smart drugs experiences. My email is jesse@smartdrugsmarts.com. Smart Drug Smarts has a mailing list. We call that the Brain Breakfast. Comes out weekly or thereabouts. Actually been doing a pretty good job the last couple of weeks. The last one was a post about memory in the animal kingdom, as a matter of fact. Something I was thinking about after last week's Dognition episode. You can sign up for the Brain Breakfast if you have not yet done so at smartdrugsmarts.com/newsletter. Might have a little bit of a preview coming up for next week's back to school episode in the next Brain Breakfast to go out. We've been gearing up on the Axon Lab side of things for a back-to-school sale. Going to do something pretty awesome. Cut all shipping costs on all domestic shipping for subscription purchases. If you're a student, you want Nexus Mitogen delivered straight to your dorm room month in, month out. Don't have to worry about reordering. Don't have to worry about postage. Until September 7th, we've got this back-to-school offer going on. SmartDrugSmarts.com/back-to-school. On the Vitamunk side of things, if you hear this episode and you're interested in fighting inflammation, but you want to wait and see the results of the human studies before psychedelics become your anti-inflammatory go-to, Vitamunk has a joint health supplement called VibraJoint. A lot of different anti-inflammatory ingredients: pycnogenol, black pepper extract, glucosamine, and the extract from Cissus quadrangularis. Worth checking out all of what Vitamunk has to offer, which you can do at vitamunk.com, or if you want to get onto their launch list as they launch new products, smartdrugsmarts.com/launch.
Smart Drug Smarts.
So I mentioned last week that this week would be the first follow-up in a series of generational interviews on Smart Drug Smarts, a father and son combo in this case. But I didn't tell you who the father was, nor the son. I already revealed the name of this episode's guest, Dr. Charles Nichols, who's actually the son of Dr. David Nichols, who I spoke with in episode number 176 about his research into psychedelic compounds, how he has been able to stay on the good side of institutional review boards, kept getting his research requests approved when a lot of other people were unceremoniously rejected, and his son and sometimes collaborator, Dr. Charles Nichols, who I'm about to be speaking with, is sticking with the family business, so to speak. A really interesting career and research path, which he will talk about in this interview. But a lot of what we're going to be discussing is one of his findings, which is not yet well known to the public, but potentially quite a big deal. That finding being that many psychedelic compounds, and some more than others, seem to have anti-inflammatory effects at low doses. Some of his research has shown strong effects in atherosclerosis and asthma. And as far as unwanted chronic inflammation in general, 50 to 60% of the population deal with this at some point in their life. So any sort of effective therapy to deal with inflammation is, of course, very welcome. And at this point, Dr. Nichols is getting a lot of the funding for his ongoing research from the NIH and private disease foundations. So important folks are sitting up and taking notice. Lots to be said here. A very interesting interview about how psychedelics continue to surprise. Let's dive in with Dr. Charles Nichols.
When I was a kid, I wanted to be a rock and roll star in a rock and roll band. Then got interested in science, and I had no intention of actually going into this field. When I was Purdue University as an undergraduate, I studied bacteria and bacterial phosphatases, and I found that really interesting. And when I went to graduate school, still didn't know what I wanted to do. I knew the kind of research that my father did with serotonin and pharmacology, and my mother was a pharmacologist. So maybe I wanted to distance myself somewhat from pharmacology because both my parents were pharmacologists. And I went into fruit fly genetics, and I studied the neurogenetics of how fruit fly eyes develop for my PhD dissertation. And then I never wanted to see another fruit fly again after I got my PhD.
Yeah, you've seen one fruit fly swarm, you've seen them all.
Oh, yes. If there is a fruit fly hell, I know where I will be going.
Nice.
But I wanted to do something more interesting for my postdoctoral work, and I wanted to do something in mammalian systems and behavior. So I looked around a little bit, interviewed in a lab doing some dopamine pharmacology, and just on a random happenstance, I happened to see an advertisement for a laboratory at Vanderbilt University to study the molecular genetics of serotonin system. And it was an investigator, Elaine Sanders Bush, who my father had collaborated with, and I knew the type of work that she did with serotonin. And I thought, oh, that could be interesting. So I sent her an email if she was interested in having me interview for a postdoc, and she contacted me back saying, well, I'm sorry, I don't know why I was listed on there. The position's not available. I don't have any funding for it. So that was that. And then it was about a month later, I got an email out of the blue from Elaine saying that if I was still interested, she'd love to have me down for an interview. So I went down, I interviewed. She's a pioneer in the study of serotonin 2 receptors and had done a lot of work with psychedelics in the 80s and was really instrumental in identifying a lot of the early signaling pathways from the receptor. And she had this project in mind to look at the genetic effects of psychedelics. We called them hallucinogens at the time in the brain. And she wanted somebody to come to the lab who is an expert in genetics to really look at the brains of rats given LSD to see how it affects gene expression. Oh, that's really interesting. That's cool. So I accepted the position, moved to Nashville, and it was a couple weeks into my postdoc where she came up to me, and you have to know Elaine. She's a very red-headed firecracker lady from Kentucky with a southern drawl. She came up and said, I was looking at your CV again. I see that you were Purdue, and I know Dave Nichols at Purdue. Do you know him? And I said, yeah, he's my dad. And she just went, I laughed. And from then on, she called me Dave in the lab. That's kind of how I got into the field. It was really not intended at all. And just through happenstance that I happened to see this ad and that she had called me back. And for whatever reasons that may be, I've now been in the field for 20 years now.
One of the things that seems most interesting about psychedelics is the potency at really small microgram level amounts, and yet they're having effects on serotonin receptors. What does this say about the potency of serotonin itself? I mean, is it like serotonin but a bazillion times more potent, or is serotonin itself just an extremely potent molecule within the brain?
There are differences. Serotonin itself will bind to, in mammals, at least 14 known receptor targets. And its affinity or stickiness for each of these is vastly different, where for some receptors, you have to have a lot of serotonin. For other receptors, just a little bit to activate them. For the serotonin 2 receptor, serotonin is fairly potent at it. But there's a key difference between serotonin and psychedelics. And when you activate the receptor with serotonin, even the high levels of serotonin, you don't get psychedelic effects. So you can artificially increase the amount of serotonin in a person or an animal, and it's not psychedelic. It has these other effects that are known as a serotonin syndrome. And some people will get that if they say overdose on an SSRI and they're taking maybe a monoamine oxidase. But they're very different symptoms.
Syndrome never sounds good, so I assume there's negative consequences there. What actually happens?
There's hyperthermia, the body heats up, tachycardia, there's a lot of physiological effects. Not many people will die from it. They feel so horrible, they'll go to the emergency room, and there's the classic hallmark symptoms of serotonin syndrome, and so they just basically wait it out and they recover. With psychedelics, it's not that they're more potent or less potent at the receptor than serotonin, but they activate the serotonin receptor differently. Mm-hmm. So when serotonin binds, it causes this cascade of enzyme activation downstream of particular sets of enzymes, and that ultimately will change how that neuron is firing in the brain network. And what psychedelics do is we believe that they recruit different enzyme cascades than serotonin that lead to both the psychedelic effects and the anti-inflammatory effects.
That might be kind of a good segue into your work looking at the anti-inflammatory effects of psychedelics, which I think is something that we can safely assume most people will not have heard anything about. How did this come to your attention? What made you look in this direction at all?
It was really an accidental discovery. I had recently established my laboratory here at the university, and we were looking to identify some cell culture assays that we could look at the gene expression response to psychedelics like LSD and DOI, because one of my main interests is to identify these different enzymes from the activated receptor with psychedelics and serotonin. And it's kind of a hassle to always have to treat an animal and take the brain out and analyze it. So we wanted to find some tissue culture models that we could do in petri dishes. And we looked at a lot of different assays to see if we could find one that psychedelics had an influence on. And one of them was brought to my lab by my postdoctoral researcher at the time, Beng-Ning Yu. It had experience studying atherosclerosis in blood vessel cultures in a petri dish. And so we took this assay, we put in this molecule that causes inflammation, and then we added the DOI to it. And much to my surprise, we very potently blocked the inflammation produced by this molecule called tumor necrosis factor alpha. At first, I thought that the doses were wrong, things were not right. So he repeated it again, got a beautiful dose response curve. I did it. And then I finally convinced myself that we were really onto something here. So it was first in muscle cells from aortas that we looked at. And from that, we looked at different types of cells and different psychedelics and found that they were all anti-inflammatory. Even LSD, which was the least potent anti-inflammatory that we found, was still as potent as a typical corticosteroid at its receptor target.
That's interesting that LSD was the least potent given its sort of famous levels of potency for psychological effects.
Right. And since then, we've looked at several different structures to try to identify what the pathways are. And I've convinced myself that the psychedelic behavioral pathway is different than the anti-inflammatory pathway because we don't really see a correlation between the potencies of psychedelics in humans to produce these psychoactive effects and the potency of the drugs in animal models to treat inflammatory disorders.
Inflammation is there for a good reason. If you have an acute injury, it's very, very helpful. But of course, one of the problems with everyday life nowadays is that people often live in constant low-grade levels of inflammation that never fully goes away. What do you see with these anti-inflammatory effects as potential therapeutic options?
So what we sort of have envisioned as a therapy is that the really ultra low doses that we're using, in some cases we predict about 100 times less than you would need to just get a minimal behavioral effect, that it's not going to be engaging receptors enough to have any pathology, but there will be sort of this low level counteraction against inflammation. And diseases like rheumatoid arthritis, asthma, depression, potentially even diabetes and metabolic disorder that we have some data for that just really involve this chronic, persistent, low-level inflammation. I think that this will be really effective as a therapy for.
And this would be at a totally subperceptual dose range. So, for example, somebody microdosing mushrooms might be a way to accomplish this?
That could be one way to put it. It would really depend on the molecule. With something like LSD, I think you're going to be in the same range of perceptual as anti-inflammatory. But some molecules like DOI, which are 100 times more potent, for reasons we don't yet understand, I think are going to represent the best candidates because they're really not going to have any behavioral effects at all that are noticeable.
And DOI isn't one that we've discussed before on this show. Can you define that? Because I think a lot of people probably won't have heard of that one.
Right. DOI is a chemical that overall in structure is related to mescaline in that it's a phenethylamine. So it's related to amphetamine in structure. And it was first synthesized by Sasha Shulgin back in the 1970s. And it's similar to the street drug that was called STP back in the 1960s. We call that one DOM, and this is DOI, where it has an iodine instead of a methyl group, but it's very closely related to amphetamine, but it's not an amphetamine. It doesn't have any dopamine behaviors or components that it mediates. And one of the reasons that it's not really widely known or, say, abused recreationally is that in humans, it's a 24 to 30-hour trip.
Oh, wow.
It's not something for the casual user to, say, go to a concert and take some DOI and come down 2 to 3 days later. One of the great uses for it is that it's the only selective drug that only targets the serotonin 2 receptor. That something like LSD will hit 8, 10 different receptors. Psilocybin will hit 5, 6, 7 different receptors. And DOI is only specific for the 5-HT2 receptors, the target of the psychedelics. So it's the most widely used chemical reagent that, as pharmacologists, can use to study the 5-HT2 receptors apart from all the others. And that's why we use that instead of, say, LSD, because it gives a much, much cleaner result in the experiments.
What is the difference in the half-life or binding times for the different chemicals within the receptors? Is it just sort of the chemical stickiness of a given compound in a different receptor, and that's why mushrooms might have a 6 to 8 hour perceptual timeline, whereas LSD is longer and DOI is much longer than that?
That's been a mystery, is why LSD lasts 8 to 12 hours when pharmacologically it's a really poor ligand. It doesn't bind to the receptor as tightly as the others, and it doesn't activate it as much. But there was a recent publication from a couple months ago that showed that there's a little lid on the top of the receptor that traps the LSD molecule in there. So it doesn't come off of the receptor. And because it sticks on there, it keeps signaling, it keeps the receptor active longer than, say, something like psilocybin, which is going to go into the receptor, activate it, and pop back out. It's not going to be stuck in there. So that could be one of the bases for why LSD lasts for so long and psilocybin doesn't. For DOI, that hasn't been looked at yet. Some of the other things to take into account are metabolism, that different classes are metabolized by somewhat different enzymes and can last longer. The half-life for DOI subcutaneously in a rodent is about 5 hours. So if you inject it, it lasts about 90 minutes. If you put psilocybin or LSD into a mouse, the half-life is only 10 minutes because it just gets metabolized. So you can have variability in the human populations as well, depending on how active their metabolic systems are. So it's a combination of how active is a person metabolizing the drug and its stickiness to the receptor, and then how well it's activating the receptor.
Is there anything that a person can do to loose these molecules from your receptors quicker if you want to? If you need to pull the ripcord and get out of a psychedelic trip more quickly, is there anything other than waiting it out?
It's not like you can go for a jog and you get rid of all the LSD in your body now. So there are a couple different enzymes. There are monoamine oxidases, which are free-floating enzymes. There are enzymes called P450s that reside in the liver that are responsible for the detoxification of most chemicals in the body that also metabolize. And drug-drug interactions are usually one drug can either upregulate or downregulate the expression or activity of one of these P450s. A good example of that is, say, codeine. There are people who metabolize codeine very poorly, and those that metabolize it very rapidly into the active drug morphine, based upon the nature of their P450 metabolic enzyme. You could conceivably figure out what would upregulate a metabolic enzyme and eat that food or take that drug. And then if you took the corresponding psychedelic that was metabolized by that, you would metabolize it faster. But we don't know which ones metabolize the psychedelics. Those studies haven't been done yet.
So to get back to the therapeutic uses for the anti-inflammatory effects, where are things at in the potential drug discovery or therapeutic recommendations? I suffer from allergies in certain parts of the year. I had asthma when I was a kid, so my ears definitely perk up when I hear about anything that could dampen down a body's response like that.
We have some intellectual property and patents that have been issued and applied for regarding several aspects of inflammation and the serotonin 2 receptor activation. In the asthma studies, we've found that we can both prevent the development of allergic asthma and rescue asthma that's already present. Mm-hmm. We're now working towards developing a clinical trial protocol to hopefully within the year or so test DOI in a small cohort of human asthma patients for efficacy. And that's kind of our goal is within the next year to have that data to demonstrate proof of principle in humans.
Do you see any potential roadblocks, or do you think that this will work? Is there cause for optimism?
I think there's a lot of cause for optimism. The one main potential roadblock is that the road to market is littered with thousands of drugs that were successful in curing diseases in rodents, but not humans. So it's really, can we make the leap from rodent to humans? Because all of the anti-inflammatory effects that we've looked at, we've found for diabetes, metabolic disorder, high cholesterol, asthma, inflammatory bowel. There's efficacy in both rats and mice and tissue cultures of rats and mice. So it works great. I have no doubt if we wanted to cure these diseases in your pets, guinea pigs, it would be awesome. But can we make the leap to humans? That's the big unknown. And I'm fairly optimistic that we can, because in a couple of the cell lines that we've tested, they've been human cells. And we've shown that the anti-inflammatory effects are there in human cells. So I'm fairly optimistic on that. And just from anecdotally, I, as you can imagine, get all sorts of emails from people that have read my work from all walks of life.
Yeah.
And I have several anecdotal reports of people who have conveyed to me their experience with their inflammatory conditions, and after using psychedelics that it will go into remission or be treated better than the therapy that they're taking. In some cases, the therapy is not working, and they say, I've tried to use it in this way and I'm symptom-free for a month or two and it's wonderful. And so I have several anecdotal reports, although those have to be taken with several grains of salt. But I'm really optimistic.
One question that comes to mind is people have been doing psychedelics for a long time. People have had inflammatory issues for that entire time. Are we just noticing this now? Even anecdotally, if there were really something there that, where there's smoke, there's fire, we would have maybe been seeing the smoke signals before now.
Yeah, I think a lot of what I'll receive, for example, will be an email from somebody who says, you know what, I always noticed that whenever I took mushrooms, that my asthma seemed to get better for a week or two or something like that. So I think it's been there, but it's been written off by the people who have noticed it. And people don't use psychedelics on a daily basis.
It is unfortunate, too, because when you hear something from somebody that's talking about, well, I did some psychedelics, and then, well, the easy dismissive thing to say is, well, that person was tripping out. Of course, they thought, fill in the blank.
Right. So you've had these people who use them not on a daily basis or a therapeutic basis, but maybe once a year, twice a year, they go to a concert or something and, my asthma got better for a couple of weeks, and then they just write it off. So I think there's been a low-level signal that just really hasn't been picked up on. I think the therapeutic strategy going forward is going to be really these low-dose therapies on a regular basis for these long-lasting effects.
What does that mean for a compound that's been synthesized like DOI? I don't know what university Sasha Shulgin was at when he created that originally, but as far as something like this entering the big pharmaceutical pipeline of being a prescribable drug, What happens? What are the intellectual property ramifications?
Yeah, with DOI, it's different than most drugs. Most drugs are made in a university. The IP is filed on the structure of them, and then they go to market and they make their money. DOI was synthesized by Sasha when he essentially was working out of his laboratory at his farm. So it's been in the public domain for the last 40 years or so. So it's not a patentable thing in terms of structure. But the normal pipeline would be to demonstrate safety and efficacy to get FDA approval. And to demonstrate safety, we have to do several experiments in rodents, increasing the dose, finding out what the toxic dose is, how it's affecting the different tissues. So we have to put DOI through the same rigorous preclinical testing that any other drug would have to go through. Psilocybin did not really have to go through that because it had been used for centuries by indigenous populations and have a demonstrated safety profile. So they were able to do that with psilocybin. But with DOI, we're currently in the process of doing those types of experiments to get the FDA approval to take it into humans.
I'm always fascinated with the concept of the pharmaceutical index, which is a term I try to throw in every 5 or 10 podcast episodes, just because I think it's one that most people haven't heard of, but it's such a valuable thing to be aware of like that ratio between an effective dose and a lethal dose. And I know that it's something incredibly high in psilocybin. It's like 300 to 1 or something.
Oh, yeah.
What do you see as the pharmaceutical index in some of these other psychedelic compounds?
I think we're looking at two different indexes. One is sort of the LD50. Half the people die. And that's huge for these drugs.
It would be like you would have to eat a garbage bag of mushrooms.
Yeah. You can take massive amounts and you're not going to be doing any physical harm to your brain or your body. People have in the past anecdotally accidentally sniffed a line of LSD thinking it was cocaine or something like that. And they go unconscious, sometimes for several days, but they recover. And it's not toxic at that level. I think in humans, you'd have to really work hard to get a toxic, lethal dose of most of these types of drugs. The different one being the one that's now known as a 25I or N-bomb. That does have toxicity associated with it. But all of the others, that's not really an issue. What is the issue is what's the dose of where you're starting to get perceptual changes.
Sure.
And so the therapeutic index that we're working with DOI and these other drugs is what's the maximum dose isn't what's going to kill you, but when you're going to start hallucinating. And so that's what we're in the process of identifying now for DOI and a couple other of these chemicals. And there is already an FDA-approved psychedelic drug on the market called lorcaserin that is marketed as a selective serotonin 2C agonist, but it's not very selective. And if you take just a little bit more than prescribed in humans, there was a paper that they had people who were experienced on LSD and heroin and multi-drug users, and they gave them Belviq. And at about 3 to 4 times the normal dose, it was producing a full-on acid trip. So there is an FDA-approved psychedelic on the market with a much lower and narrower therapeutic index based upon the behaviors than I think DOI would be on the market. So I think that elephant is out of the room now.
That is a little-known fact. Hasn't made it out into circulation much.
I used to get questions all the time. You think the FDA will approve a psychedelic? And they've already done that.
How widely prescribed is that compound? Is that something that sees the light of day a lot?
It is for essentially morbidly obese individuals that is effective in suppressing the appetite for people not to eat as much. And it's effective in about half the population, probably due to metabolic genetics, where they lose a significant amount of body mass, of BMI, and they will maintain that through activation of the 2C receptors. But its activation of the 2C receptors has been shown to also produce anxiety. So the psychedelic trip that these human subjects in the research study had was unpleasant. And for them, it was psychedelic and psychoactive, but it was very unpleasant. So I think it's a combination of you're activating the C receptor and bringing in anxiety with the A receptor for the psychedelic effects, and they just don't want to take it again. So it's not got a reputation for being something that a trip that you want to have.
So self-reinforcing and not wanting to take too much of a dose, which is probably a very good thing as far as not getting prescription users into trouble and also getting doctors more comfortable in actually prescribing these sorts of compounds. How have you found the general political climate in doing these sorts of studies? Have you been getting the evil eye? Is there still a taboo?
When I first got into this field 20 years ago, my father told me it's the kiss of death. If you go into the field of psychedelics, that's it. Nobody will fund you. You won't be taken seriously. But I did it anyway. And I think it's the approach that I've taken to study molecular mechanisms, what's going on, and how these drugs activate the receptor in the brain, because to me it's fascinating that you can activate these receptors and have people report that they're in a different reality. I think that they're really fundamental to our understanding of consciousness and mental disorders that change our consciousness. So I've approached it really from that level and have been successful with NIH funding to understand really the biology of the serotonin 2 receptors and its relationship to normal cognitive processes, and by extension, developing therapies to treat diseases are similar to some of the effects of psychedelics like schizophrenia. Mm-hmm. So I've had a lot of funding from NIH to study that. And then when we discovered the anti-inflammatory effects, it's novel because we're not inducing these CNS effects, and it's in the periphery. So I've had success from several different funding agencies, NIH included, some small foundations as well, that aren't dedicated to studying psychedelics like MAPS or Hefter. So scientifically, I think it's been not as difficult as I originally thought it was going to be because of the questions that I've been trying to study and answer.
For the anti-inflammatory effects, it seems like such a wide swath of society is affected by something that falls under that umbrella. I mean, do you have any numbers there as far as what proportion of the population deals with inflammation issues?
Well, the leading cause of death in this country, one of the leading causes, if not the, is cardiovascular disease, atherosclerosis, coronary artery disease, diabetes, metabolic disorder. And if you have, say, 200 million adults, 100 million who are affected, who could potentially benefit, that's 30 to 50% of the adult population, which is a huge number. And we've got animal data for each of those indications that this therapy might be successful. And I think that the anti-inflammatory effects are also relevant to the antidepressant effects that have been seen in the clinical trials. And that's one of the things that we're starting to look at now in some animal models.
Can you dig into that one a bit? I would love to hear about the potential crossover between the psychological effects and the anti-inflammatory effects, how those tie together.
So I think in depression, there's a large percentage of people who are depressed that have inflammation associated with the depression. And so that's been a really hot topic in neuropharmacology. The last 7-8 years has been neuroinflammation as related to depression, bipolar disorder, schizophrenia, that these people have activated microglia in the brain, which are the responsiveness cells to inflammation. There's increased inflammatory markers. The brains of these patients look inflamed. And so the theory has been if we could treat the inflammation, then we can alleviate the depression. So there have been several clinical trials using anti-inflammatories to treat depression. And they've shown that, for example, depressed patients who show inflammation in the brain before taking a selective serotonin reuptake inhibitor like Prozac, after they have been treated and their depression goes away, the inflammation goes away as well. So it's still kind of a chicken and an egg.
Yeah, sort of a vicious cycle there.
Right. So my current hypothesis is that the psilocybin in the clinical trials is having an anti-inflammatory effect within the brain, that when somebody goes into these trials, they're depressed, they take the psilocybin, they have their mystical experience that Roland Griffiths has spoken about that seems to be integral to them recovering from the depression. But then ketamine does the same thing, that treatment-resistant depression, you give the patients ketamine, they're better, but only for a couple weeks. And why is that? Where psilocybin seems to be effective for a year and a half out without really any decrease after a single dose. Mm-hmm. And so what I think is that mystical experience that you're having, regardless if it's ketamine or psilocybin, is getting you out of the depression. But unlike ketamine, with psilocybin, you're treating any underlying neuroinflammation. So you're normalizing the inflammation and the brain doesn't get sucked back into that depressive state after a few weeks. So I think that it's a really integral part of the antidepressant effects of psilocybin, but that's something that we have to validate experimentally.
Problem is sort of try to dissect what's actually happening there, since these compounds are affecting both psychological levers and neurophysiological levers.
Right. And if we could separate out the inflammation from the psychedelic effects, which I believe we will be able to sooner than later, and if you give a psychedelic non-anti-inflammatory versus a psychedelic anti-inflammatory, and then look to see in head-to-head trials which one will be more effective than the other. And I would predict that the psychedelic anti-inflammatory is the one that will be effective 1 year out, 2 years out, whereas the non-anti-inflammatory psychedelic may only be effective for a few weeks.
And is that a study that you're actually planning to conduct sometime in the reasonably near future?
Oh, I would hope so. We are actively dissecting out the pathways of these different classes to try to identify psychedelic anti-inflammatory versus the psychedelic non-anti-inflammatory and vice versa combinations. So hopefully within the next couple of years, we'll be able to test some of these hypotheses. That's one advantage to having a chemist in the family. I'm fortunate enough to be able to have a large library to test the structure, function, activity of these molecules basically at will without having to sort of say, well, what happens if we have this molecule and we remove this? We don't have to go out and get it made because I've already got it.
Early in the conversation, we were talking just a bit about the epigenetic changes that can happen from psychedelics use. It's funny, though, the first thing that I really remember hearing about psychedelics was Nancy Reagan era scare tactics. They'll screw up your genes. This idea that psychedelics had actual germline changes, which of course isn't really what happens, but epigenetic changes are happening all the time. What do we know about the epigenetic changes that come along with psychedelics exposure?
We've looked at chronic and acute. I'm not going to go into the chronic too much. That's a model of psychosis that my father developed, where you give LSD every other day to rats, and after 3 months, they essentially go crazy. And it's all in the timing. It's every other day. If we do it every day or every third day, it doesn't work. But every other day, and it's only with LSD. It's not with any of the other psychedelics.
And they never bounce back from it.
No, they don't. We stop the drug and they stay crazy.
What defines a crazy rat, by the way? If you met a rat, how would you know he's crazy?
Very aggressive, hyperactive. They startle a lot. We try to pick them up. They jump. They try to bite. The normal rats, they don't really do that. They're very friendly.
Okay.
But they had all the similar symptoms that if you give a rat high doses of methamphetamine, it acts very similar. And when we looked at the brains of those rats, we found that there was a very significant enrichment of genes that are implicated in schizophrenia and bipolar disorder that were dysregulated. So that was sort of a moderate, low, chronic, every other day for 3 months.
That's fascinating that it was not every day and it was not every third day, but something about hitting that exact ratio of days.
Right. And that's not how people normally take these medications, but it was following up on some observations that my father had made on his rats over several years. And lately we've been looking at more of what are the effects of a single acute dose. And we've looked at DOI and LSD, and we've been treating the animals and then 45 minutes later dissecting out the brain. And we've figured out how to sort out the different cell types from one another. And we've identified that there are about 5% of the brain neurons that are directly responding to psychedelics, that most of them don't respond. It's about 5%. And we've been able to purify out this 5% of neurons, and I call them the trigger population, that the 5% of neurons that activate directly with psychedelics, that their activation then sort of propagates the signal to the rest of the brain.
Are these 5% localized in any particular brain areas?
They are localized to a few different brain areas. To the medial prefrontal cortex, we see a lot of the activated neurons, and we also see a lot of them in a structure called the claustrum. And the claustrum is one of the most highly interconnected brain areas, but it's also not studied very well because it's very hard to get to. It's like a little ribbon that runs from the front to the back of your brain that's connected to almost every structure. We see a high level of activation in that structure and the medial prefrontal cortex, which is sort of the executive part of your brain that most of your cognitive abilities reside in.
Yeah.
And we've been able to look at those 5% trigger population neurons and do gene expression profiling on them and see that there are a lot of early responsive genes that become activated. There are some circadian rhythm genes that become activated, but how they're activated at the genetic level changes from one brain region to another. So when we compare what would otherwise be the same neuron type from the medial prefrontal cortex to the somatosensory cortex, which is responsible for movement, they're activated differently. Different types of cells are activated, and the types of cells that are activated have different genetic responses. And that was something that wasn't really known before. I think that may help to explain some of the connectivity data that the Imperial group in the UK is seeing, that you're strengthening some connections and weakening other connections, but overall increasing connectivity, that within the different brain areas, we're affecting these cells at the molecular and the genetic level differently. And so you can't really look at it as this brain area is becoming more active and talking with this one. But within that brain area, you're getting cells that are excitatory and cells that are repressive, both becoming being activated in somewhat different balances. So you look below the surface, it gets even more complicated and more questions come up.
Does there seem to be consistency maybe between people or even between the same person on multiple doses as to these levels of activation? Or is there somewhat of pulling the lever on a slot machine and you get different activations every time?
I think that gets back to the old adage of set and setting, that the state of your brain is really at any given time defined by your mood, your setting, where you are. So the actual state of your brain is going to be different from, say, one use to another use. And that, I think, underlies a lot of the variability of why one experience, say you're at a Grateful Dead show, is going to be different than you're at your grandmother's funeral if you're taking LSD or something like that, because the states of your brain before are going to be somewhat different because of the activity, the inputs coming in.
And how long do these induced epigenetic changes seem to actually last? How long do they persist, I assume, beyond the actual period of psychedelic effects and perceptual changes?
For the chronic studies, where we gave them LSD for 3 months, we did the gene expression studies 4 weeks or 1 month after the final LSD dose. And the changes were really profound at that point. So I think that within that model system, the changes are fairly stable and persistent. We haven't looked at the length of the changes in the acute studies. We've looked to 5 hours out. Most of the genes are back to baseline level by 5 hours, but there's some that are still maximally induced at 5 hours. So it could be that somebody takes a single dose, they're inducing a lot of genes, changing the function of the brain cells, and then after the course of the trip, the genes go back to normal for the most part, but there's a few genes that are still not back to normal that are giving a slight change to the brain neurons and how they're functioning. And so if they take another dose, say a day or 2 or 3 days later, they're sort of bootstrapping on that and recruiting additional genes.
I forget whose study it was, but the finding that psychedelics are, I think, still the only known way of reliably changing one of the 5 basic personality characteristics, and it increases openness.
Right. I think a lot of those studies have been out of the Franz Wallenweider's group in Basel in Switzerland. They've been doing a lot of studies on personality with that. But yeah, that could be that maybe some of the genes after even a single dose, they might permanently be altered. And we can't say that they're not. And that's at the expression level. And we're just now beginning to look at epigenetic changes and chromatin structure to see if those are really playing an issue or not. But it's quite possible that certain genes come on after a single dose that just don't go off again. And that could be underlying some of the positive therapeutic effects.
Do you suspect that you'll see those sorts of persistent changes even in the ultra-low doses that might be used for anti-inflammatory effects?
I think they're engaging 2 different systems, and you need more of the drug to engage the psychedelic system than the anti-inflammatory system. So the low-dose anti-inflammatory, I don't predict, would be associated with these CNS gene expression changes, but the experiments just haven't been done yet.
SmartDrugSmarts.
So a very big thank you to Dr. Charles Nichols for taking the time for that conversation. Really interesting when a finding like this can sort of be hiding in plain sight for a long time before anybody notices it in a rigorous way like this, before it becomes more than anecdote. Like Dr. Nichols said, the fact that most people who use psychedelics use them so infrequently that it's kind of hard to pick up the pattern, the signal in the noise, that this anti-inflammatory effect might exist. You're only getting any sort of signal maybe a couple of times a year, and compared to the much more overt effects of major psychological distortions while a person is on psychedelics, you might not notice that, oh, my joints feel a little bit better or my asthma went away. But that's why we have science, so better late than never on this finding. Will be very interesting to see if this applies up the chain to humans and how this might be applied therapeutically. Keeping things a little bit on the borderlands of what you can safely talk about in polite company, we'll move on to the ruthless listener retention gimmick, where we've got a surprising mostly because it's a positive finding about how alcohol can affect the brain.
SmartDrugSmarts, ruthless listener retention gimmick.
So about a half dozen episodes ago, we had an episode about the replication crisis in science. Obviously, it's a bummer for all involved when a finding cannot be replicated. Were things done correctly? Was it just a fluke finding? Or possibly, were the conditions that triggered the effect more specific than the researchers had assumed? Might it be something where an effect can be seen in laboratory conditions, but it doesn't necessarily translate out to the real world? That does happen sometimes, so it's nice to see scientists try to replicate laboratory findings in real-world environments. And researchers in the UK recently did this very thing in a study on alcohol, looking at a positive finding on alcohol and human memory and seeing, hey, does this survive laboratory conditions and still hold true in the real world when people are drinking in the comfort of their own home? So what exactly is this about positive memory effects from alcohol use? Kind of flies in the face of common sense. Alcohol is well known as a memory disruptor, and that's generally a true statement if the information in question is something that you learned after having consumed alcohol. But the paradoxical thing here is information that you learned just before you began consuming alcohol is actually better remembered than it would otherwise be, than if you hadn't done any alcohol drinking at all. Really kind of weird, but this is what's been found in previous studies. And what scientists think is going on here is that there are various processes involved in memory consolidation. You can think about the perceptual acquisition of new information, moving that into short-term memory, starting to consolidate that into long-term memory. This means laying down of new neural connections, strengthening synapses, myelinating axons, all those things. And alcohol consumption clearly messes up the beginning part of this pathway. Basically makes the brain want to not onboard new memories into this memory pipeline process. We got some problems here. Let's handle the memories that we do have, not let any new memories in the door, and then try to sleep it off. You could kind of think of this as like a busy restaurant kitchen where each plate of food served is akin to a new memory. And the kitchen staff has a certain amount of time, maybe 20 minutes per meal, to get it prepared and get it served to the customer. They've got a system down. They can give a certain amount of loving care to each dish, but then they got to get it out there. But what alcohol does is akin to having the floor staff in the restaurant say, no new orders, we'll finish the orders that have already been placed, but otherwise time to go home. All of a sudden, the kitchen staff, they're not getting new orders coming in, and so they can lavish more attention on each of the orders that have already been placed, kind of spice them up in a way that they wouldn't have normally had time to, and those last few customers who have placed orders actually get a better meal than they would have normally gotten. That's the theory of what's going on in the brain, where we have this paradoxical finding that the memories that were induced to be formed, that sort of started this memory consolidation process before the alcohol was consumed, actually get better consolidated. So they tested this theory. They found 88 social drinkers, got them to do some learning, sent them home, and let them drink on their own recognizance. They could drink whatever they wanted, but they were just supposed to keep track of how much they'd drunk. 2 hours into their drinking, they would do a breathalyzer, and then they were tested the next day on how well those memories had stuck. And of course, there was a control group who hadn't done any drinking at all between the learning and the testing. And as was found in the lab study, the drinkers actually outperformed the non-drinkers on how well they remembered the pre-drinking learned information from the previous day. They also found that the magnitude of this effect grew the more the people drank. The total amount of drinking per participant in this study for the drinkers was 82.59 grams, by the way. The researchers do suggest doing follow-on studies that do have sleep as an independent variable. There was one night's sleep that took place between the learning and the testing, and alcohol has been shown to sometimes increase the proportion of slow-wave sleep during a night following drinking, and slow-wave sleep is known to be crucial for declarative memory consolidation. It could be that that busy restaurant kitchen analogy isn't exactly accurate. What's really happening is that alcohol is dialing up the amount of slow-wave sleep a person gets, and that slow-wave sleep is improving the consolidation of memories for the testing the next day. As of now, we don't know, so presumably more alcohol and memory studies coming in the future.
SmartDrugSmarts, where we turn information into sound, into bits, into packet data that turns back into bits and sound, and then into neurotransmitters that release funds that release mail-order synthetic chemicals that cross the blood-brain barrier to release augmented performance from your brain.
Okay, so that was it for episode number 196. Thank you for hanging around until the very end. If you enjoyed this episode, you will find everything that we talked about here up online at smartdrugsmarts.com/196. If you missed last week's episode in 195, we talked about dognition, not cognition, dognition, with a couple of dog experts. A lot of people seem to really like that episode, even though admittedly it had very little to do with the human brain. But I definitely appreciate the latitude to go off in some of these weird areas. So thank you to those of you who have said nice things about that episode. Next week, I think we'll be doing something overtly back to school in the back to school theme. I'm not going to quite tip my hand on what that is yet, but I will see you back here next week, same time, same podcast, and with that same unflagging commitment to helping you fine-tune the performance of your own brain. Have a great, safe Labor Day, and of course, stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.